Hi guys, going over chapter 7 for this recording. And actually, this chapter 7 will require two recordings. So today and also the next one would be focusing on chapter 7. Chapter 7 contains one of the most abstract concepts for the entire book, the entire Stanfield textbook, the entire course, Human Physiology. Chapter 7 is the center of attention here. Um, Although the concepts covered are abstract, they are absolutely gorgeous. They are a marvel in terms of nature, because the concept is at the center, the heart and soul of what gives rise to one type of signals, and that signaling mechanism is being used by excitable tissues, meaning tissues or cells that can generate this type of signals. You see that in neurons, you see that in skeletal muscle fibers, you see that in cardiomyocytes, you see that in autorhythmic cells, you see that in smooth muscle cells, you see that in um, pacemaker cells. All of those cells are basically utilizing the concept that we would be covering today. Um, so all the muscle tissue, um, the neurons are capable of doing this. And that concept is the capability of generating electrical signals. And when we say electrical, we're not talking about electrons, running wild like on, electrical wires. In biology, if you allow electrons running wildly, the cell will die. So the cell in biology uses another strategy to generate electrical signals without relying on electrons. It's a beauty. So, but before we get to that, we have to do some quick review from anatomy, allowing us to basically put a mindset and then present the information for me to make sure that we are on the same page. So we go back to review briefly the structural organization, meaning how nervous system as an organ system is organized. So overview of the nervous system, and briefly going over the types of cells. Again, I'm not gonna lecture on anatomy. That's not the point, and I'm not interested in teaching you anatomy again. Um, but we do have to go back and revisit some of the basics before we can talk about the functionality of this organ system. And then the gist for the lecture is the resting membrane potential. What is a resting membrane potential? And how do neurons generate and establish and maintain that resting membrane potential? And then once we define that resting membrane potential, we understand how neurons uh, sets it up and then maintain that RMPs. And then not only that, neurons can use that membrane potential as a signal. So we're going to go and talk about electrical signaling. And that electrical signaling is possible just by changing those membrane potential. So you change a resting membrane potential to a new value in terms of membrane potential, and that can be utilized as a signaling mechanism. And so that's under structure function correlates. And one of the specific example of using electricals, changing the membrane potential and using it as a signaling mechanism we're going to see that under graded potential. So graded potential is just an example of an electrical signaling mechanism for today. And then on the next recording, we're going to go and talk about the most beautiful mechanism of them all, the mechanism to generate an action potential. And so for today, we're going to end with graded potential. So that's the gist for the lecture for the first part of chapter 7. So with that said, let's get this thing started. So overview of the nervous system. So when we talk about the nervous system, the nervous system is divided into central nervous system and peripheral nervous system. And when we say central nervous system includes the brain and spinal cord. Anything else that is physically not located in the brain and or the spinal cord would be classified, would belong to the peripheral nervous system. All right? So brain and spinal cord, central nervous system, everything else, peripheral nervous system. Information that goes toward the central nervous system, they are called afferent, AFFERENT, sensory, or ascending. So they all mean information being conveyed to the central nervous system, meaning information arriving at the central nervous system. So afferent pathway, ascending pathways, sensory pathways mean the same thing. These pathways are transmitting information toward the spinal cord and then to the brain. In the opposite direction of information going, we have efferent, EFFERENT. So the trick here is A for arriving, E for exiting. So this efferent pathway and basically transmits information away from the central nervous system. So efferent pathway, descending pathway, motor pathways means the same thing, right? Descending, DESCENDING, or motor, MOTOR. So efferent pathways, descending pathways, motor pathways transmit motor commands, transmit motor decisions, which originate from the brain and or the spinal cord, and leaving them and go out to effector organs, to target organs, right? The sensory pathways, the ascending pathways and efferent pathways rely on sensors rely on peripheral receptors. They detect the stimulus or stimuli and they send it to the central nervous system. Whereas the motor pathway, they transmit information coming from the central nervous system and going toward the effector organs, right? So effector organs, we're talking about somatic or autonomic. Now, for this, we have somatic and special. You can pinpoint the exact location. When we say somatic sense, we're talking about general senses. Special sense, we have 5 special sense, right? So the 4 general sense, the 4 somatic general sense, sense of touch, sense of temperature, sense of pressure, and sense of pain. Those are the 4 general sense. The 5 special sense, sense of sight, sense of smell, sense of taste, sense of hearing, and sense of balance. Those are the 5 special sense. The visceral sense, what is unique about them, you cannot tell the exact location, right? So there's no way for you to know the discomfort, the exact location where the discomfort originates. I'll give you another example. If you have a paper cut on one of your body surfaces, you know exactly where that paper cut is. But if you have a discomfort in your visceral organs, like in your pelvic abdominal cavity, abdominal pelvic cavity, or within your thoracic cavity, good luck. Because there's no direct communication line between these visceral organs to your primary somatosensory cortex. This is the region where you know in the brain that you perceive, right? So this is basically perception, conscious awareness of a sensation. So there's a direct line of communication, for example, between your right thumb and to an area on your left primary somatosensory cortex, so that you know exactly, okay, the pain comes from a thumb, and not only that, on the right thumb, not on the left thumb. So there's a direct communication line between the peripheral sensor and the central perceptive area. In the visceral case, we don't have that connection. So let's say you have a heart attack. There's no direct communication signifying, allowing your primary somatosensory cortex about your heart. So the heart, the first order neuron that conveys sensory information coming from the heart is sharing the same second order neuron with the first order neuron coming from your left pinky. That's why when you have a heart attack, you perceive as numbness on your left pinky, because that sensation is shared by one second order neuron in the spinal cord. And that second order neuron goes to the third order neuron in your thalami, and then that information goes to the primary somatosensory cortex. So because of this lack of line of communication, any discomfort in any of the visceral organs, we do not have a clue where it comes from. So that's the difference between somatic and special sense versus visceral sense. Moving on to the motor, the difference between somatic and autonomic. Somatic means, in the case of the motor function, it allows you to control your skeletal muscles. So somatic means voluntary control. Autonomic means involuntary control. So you do not have voluntary control for your sympathetic and parasympathetic motor outputs. And under sympathetic, the effector organs are cardiac muscle, smooth muscle, glands, and adipose tissue. So add to that, adipose tissue. Parasympathetic targets the enteric nervous system, more specifically the digestive system. So sometimes some medical textbook will basically split the autonomic into 3 divisions. So the sympathetic, the parasympathetic, and the enteric nervous system, right? So in our case, let's learn it that way. I'm going to change the way this part is organized. So under Bio 32, in our case, the autonomic division includes sympathetic, parasympathetic, and enteric nervous system. So I'm going to split this internal nervous system. I'm going to put it over here, okay? Now, what would be the difference between sympathetic and parasympathetic? The sympathetic would be a gas pedal analogy. So it would prep the body for fight or flight situations, whereas the parasympathetic is the opposite. This is the rest and digest division. So after a meal, the parasympathetic kicks on, it's activated. So you're enjoying life with a cigar and all that. Whereas under sympathetic, you are stressed out, right? Your body is prepping itself to face an emergency situation. Whereas the parasympathetic, it's prepping your body to enjoy life. You are relaxed, you're enjoying the moment. And the sympathetic and parasympathetic, they do not work together. 99% of the time when the sympathetic division is activated, the parasympathetic division is suppressed. And when the parasympathetic division is activated, the sympathetic division is turned off. The only time when both divisions work as a team, sympathetic and parasympathetic are both activated simultaneously, it is during sex. So when the individual attains climax, that's the only time when sympathetic and parasympathetic comes into play together as a team to help the individual to get to attaining climax during sexual intercourse. Outside of that, these two divisions, these two autonomic divisions of our motor system do not work together. Okay. Now, we can classify neurons because we have 2 types of cells in the nervous system, neurons and support cells, the neuroglia or glial cells. So I'm not going to talk about the glial cells. I'm going to focus straight up out the bat here about the nerve cells, the neurons. And when we talk about neurons, we can classify them by using 2 classification methods, 2 classification schemes. You can classify them based on structure, the way they look, so structural classification, or you can classify them based on function, based on their functionality, so functional classification. Based on the way they look, there are 4 families of neurons, 4 types of neurons, 4 classes of neurons, 4 categories of neurons. Anaxonic neurons, meaning neurons not having an axon. Bipolar neuron, shown here. You have the dendrite and the axon dissected, intersected by the cell body of the neuron. So the dendrite and the axon are separated from one another by the cell body. The pseudo-unipolar neuron shown here, the cell body is on the side, like on the side of the road. So the axon is continuous from one side of the cell body to the other side of the cell body. So we have the peripheral axon moving past the cell body and becomes a central axon. And then the multipolar neuron, this is a prototype motor neuron. You have thousands of dendrites on one side and only one axon on the other side. So multipolar. So both bipolar and pseudo-unipolar, based on function, they would be sensory, whereas the motor is the multipolar neuron. Now, how do we classify neuron as a central neuron or as a peripheral neuron? You pay attention to their cell bodies. So I'm going to highlight here all the cell bodies in blue, the cell body here, the cell body here, and this cell body. Now you need to know the exact location of their cell bodies. If the cell body is located within the central nervous system, that neuron would be classified as a central neuron. When I say cell body within the central nervous system, I mean the cell body is physically located either inside the brain or inside the spinal cord, or part of the brain and part of the spinal cord, then they would be called central neuron. If the cell body is located physically outside of the brain and outside of the spinal cord, they would be classified as peripheral neuron. So because of that, this pseudo-unipolar neuron here, this is a typical first order neuron. This is a sensory neuron, part of the sensory pathway, right? So the first order neuron, the cell body is located outside of the central nervous system. Specifically, this cell body would be aggregated into a ganglion, like a dorsal or posterior root ganglion, right? And that ganglion, since it's physically outside of the central nervous system, it is a peripheral structure. So this first order neuron would be a peripheral neuron. Whereas these interneurons, the cell body is located in the spinal cord, so they would belong to a central neuron. Same thing here. This would be a lower motor neuron. The cell body is in the spinal cord. So this lower motor neuron would be a central neuron. So the first order neuron, which is a central neuron, sensory neuron, is a peripheral neuron, but the lower motor neuron, it's a motor neuron, it would be a central neuron due to the respective cell bodies. The sensory neuron, the cell body is outside of the central nervous system. The lower motor neuron, the cell body is within the central nervous system. That's how we know the neuron, whether it's a peripheral neuron or a sensory neuron. And that you should have learned that from anatomy. Now, let's go and talk about resting membrane potential. The plasma membrane, the cell membrane, are the phospholipid bilayer of neurons. can be used to store potential energy, can be used like a mini microscopic battery. You buy batteries to basically use to drive your toys, right? Turn on your electronic device, because the these batteries contain stored energy, potential energy. And that potential stored energy in batteries can be harvested by electrical toys, electrical devices, to basically drive them, right? So your toys can be, you can play with your toys, you can turn on your computer. That energy comes from the computer's battery. Same thing with the cell membrane. The plasma membrane is capable, and listen to this, the cell membrane, the plasma membrane, or the phospholipid bilayer, it's capable of separating charges, keeping the positive charge separated from the negative charge. All cells are neutral, meaning if you basically step away from the cell membrane in the cytosol or the extracellular milieu environment, it's neutral. Every positive charge is interacting with a negative charge. So we have neutrality, right? But only at the membrane. So the membrane is a bilayer of phospholipid, 2 layers of phospholipid. So we're going to call that those 2 layers the outer leaflet and the inner leaflet. Leaflet is spelled Leaflet. Outer, Outer. Inner, Inner. So you have an outer leaflet and an inner leaflet, right? Or the outer phospholipid layer and the inner phospholipid layer. And the charge would be sitting next to, they cover the entire surface of that inner leaflet and the outer surface of the outer leaflet. So when they are separated, so the positive charge and the negative charge would be separated, and that takes energy. So when the cell membrane is providing the energy, it will split the charge, and that energy is temporarily stored by the cell membrane, by the plasma membrane, by the phospholipid bilayer. And that's a gorgeous, gorgeous concept. The cell membrane, by splitting opposite charges from one another, stores potential energy. And when they allow the charge to come back together, energy would be released, right? So when you change the potential, you can do work. You can generate, you can use it as a signal. Okay? Now, how is it possible for the cell membrane to generate this membrane potential, this potential energy that we call membrane potential? How is it possible for the membrane to separate the charge, opposite charge from one another? Okay, so let's begin. On the cell membrane, the plasma membrane, you have transmembrane proteins. Okay, you have on the membrane TMP, little s here for plural. So transmembrane proteins, meaning these proteins are coded by genes that would have been expressed through transcription and translation, and they are translated by ribosomes that are associated with the endoplasmic reticulum, rough ER, sends it to the Golgi apparatus, and the Golgi apparatus, due to the signal peptide will basically triage it, sends it to the cell membrane, to the phospholipid bilayer, or to the plasma membrane, right? So these transmembrane proteins, they act as channels, and they're called leak channels. So they can leak potassium, they are permeant to potassium, or they can leak sodium. So we have two distinct type of transmembrane protein. One is specifically leaking potassium, one is specifically leaking sodium. What do I mean when I say leak? Meaning they are open 24/7. These transmembrane protein, they are channels, they are cation channels that have a pore, that have an opening, a passageway, a tunnel, which remains open 24/7, right? And they're located at the cell membrane. They're located at the plasma membrane. They're located at the phospholipid bilayer. Okay, now, let's start at the beginning. So the beginning is we have no membrane potential, right? So 0 millivolts. So there's no, your battery is dead. So when your battery has no more power, it will be displaying 0 millivolts. So when you're buying a battery, like a 9-volt battery, that battery is basically has energy that we express as volts, right? It has 9 volts. Batteries, most of the batteries have 1.5 volts, right? So you buy a battery, it tells you how much power it has, how much energy it has. So in this case, in this membrane here, when there's no, at the beginning, there's no energy because the membrane hasn't set that membrane potential yet. So it's at 0. So we have to start somewhere. So at the beginning, there's nothing. So with that said, so that's panel A, right? Okay, now let's begin. In the plasma membrane, you have these leak channels, like I mentioned earlier. So we have leak potassium channel, and we have leak sodium channel. Now, there are more leak potassium channels open than leak sodium channel open. In other words, the cell membrane is more permeant to potassium than to sodium. More specifically, 25 times more permeable to potassium than to sodium. So if you have channels open, so what happens when the cell, right? So you will have potassium is leaving because potassium is much more concentrated on the inside. So you have a potassium efflux. You also have sodium coming in. So sodium influx. The problem is you have much, much more potassium leaving. The cell is letting much, much more potassium leaving than allowing sodium to come in. So the cell is losing positive charge faster than it gains positive charge, right? So when sodium comes in, the cell is gaining positive charge. But when potassium is leaving, the cell is losing positive charge. So over time, if the cell is allowing much more potassium leaving than sodium coming in, at the tune of 25 times more potassium leaving than sodium coming in, it will accumulate, it will lose much more positive charge inside the cell. It becomes more negative than outside the cell. Now, this is very misleading. It doesn't mean that there's negative charge here. The negativity here only means that now outside you have more positive charge than inside. So inside you have a tiny plus sign, but out here you have a massive plus sign, right? Because there's more potassium leaving, okay, than sodium is coming in. So inside the cell becomes more negative, meaning it has less positive charge due to the massive efflux of potassium at the tune of 25 times more potassium leaving than sodium coming in. Now, that's due to concentration gradient. That's due to the gradient of the chemical. So this is a chemical gradient. So panel A and panel B shows you this, all of that is driven by the chemical gradient. So delta C. The concentration gradient of potassium drives potassium efflux. Concentration gradient of sodium drives sodium influx, right? So that's the chemical component. That's a chemical gradient. So chemical for potassium and sodium due to the gradient of their concentration. It would set panel B. And once that happens, we see something emerging. You have now the appearance of opposite charges. Now you have an electrical component, electrical force. So opposite charge attract. So once inside the cell is more negative, it's going to attract the outer positive charge to come into the cell. So in other words, the negativity inside the cell will slow down the potassium efflux and will increase the sodium influx because opposite charge attract one another. So potassium is positive. So if there's an increase in negative charge inside, then it will slow down potassium efflux because of the electrical force will basically oppose positive charge from leaving the cell. Simultaneously, that negative charge will attract the positive charge from the outside of the cell to coming into the cell. Because of that, this electrical force slows down potassium efflux, increases the sodium influx, right? So ultimately, this is the electrical force. Now you have the chemical force favoring potassium efflux, chemical force favoring sodium influx, electrical force favoring sodium influx, it also favors the slowdown of potassium efflux. And ultimately, potassium, it will be forced to be going into due to that negative insight. And then at equilibrium, there's no net movement of charge across the cell membrane, and we have potential energy minus 70 millivolts. And that's the amount potential energy is expressed as minus 70. The sign minus means inside is negative, outside is positive, not inside the cell. Along the surface of the inner leaflet of the membrane is negative versus the outer membrane is positive. If you drift away from the cell membrane out here, it's completely neutral. In the cytosol, it's neutral. It's only at the two leaflet of the cell membrane where you have charge separation. And that's your stored energy. That's your potential energy, the ability to do cellular work due to that potential energy. And at equilibrium, it is at minus 70 millivolts. Each ionic species has its own equilibrium potential. Sodium is plus 60 millivolts. Potassium is at minus 94 millivolts. Now, since the membrane is more permeant to potassium, that's why the resting membrane potential is at minus 70, is closer to potassium than to sodium, due to the fact that the cell membrane is more permeant, is more favorable to potassium flux than sodium flux. Because of that, the cell membrane potential at equilibrium, it sits at closer to minus 94, which is minus 70, than plus 60. Now, if you let this cell on like this forever, it won't, that membrane potential minus 70 millivolts would disappear, would dissipate. Because ultimately, the concentration become equals inside and outside for both potassium and sodium. And the membrane potential will become zero back here. So ultimately, sodium will respect the electrochemical gradient, potassium will respect its own electrochemical gradient, and ultimately there's no net exchange, and that potential drops to or goes back to 0 millivolts. So the cell needs an additional mechanism. In addition to the leak channels, the cell will use energy to make sure that that minus 70 millivolts remains minus 70 millivolts. And that's exactly what happens. Introducing back the star of the night, sodium, potassium, ATPase. So this is a pump, require energy. So this is an active transport mechanism. More specifically, this is primary active transport. It consumes ATP to kick out 3 sodium and bring in 2 potassium. So in other words, it will establish this membrane potential. And not only establishing it, it will maintain the membrane potential at rest at minus 70 millivolt at the expense of cellular energy / ATP. So sodium, potassium, ATPase, or sodium, potassium pumps establishes the resting membrane potential and maintains that resting membrane potential at minus 70 millivolts for neurons. So each cell has their own resting membrane potential. That resting membrane potential can fluctuate anywhere from minus 5 millivolts all the way to minus 100 millivolts. And neurons have minus 70. And later in the semester, we're going to see cardiac muscle, contractile cardiac muscle cells or cardiomyocyte. The resting membrane potential sits at minus 90 millivolts. So depending on the number of leak channels they have, depending on the number of ATPase they have, the resting membrane potential will change. And that's cell type dependent. But every single cell in your body has a resting membrane potential, has potential energy to do work due to its ability to separate opposite charge from one another across that phospholipid bilayer, right? So I made sure that I wrote down the terminology for you. So we have the inner leaflet of the plasma membrane, and you have the outer leaflet of the plasma membrane, right? So the inner leaflet is negatively charged, outer leaflet is positively charged. That's why we have the negative sign here, right? So all these negative signs here, that's on the inner leaflet of the membrane. It doesn't mean it's inside the cell, it's just the lining of the inner leaflet. And it's not about negative charge, it's just less positive charge compared to the outer leaflet, right? So in order for us to express that, we write down negative, but it doesn't mean that you have actual negative ionic species sitting there, right? There's no negative ionic species sitting there. It's just the difference between positivity. So the inner leaflet has less positivity than the outer leaflet. That's the true meaning between the negative sign shown on all of the figure in all of the medical textbook. And nobody will tell you that, right? So this is basically insider knowledge here. And I'm a neuroscientist, so my training is actually in the field. So I'm a little bit biased when I'm talking about the nervous system and the cardiovascular system, where it involves excitable tissues. So this is my passion for doing this, and I've been doing this for more than 25 years now. So there you go. Okay, with that said, let's get the ball rolling. Let's keep on moving. So now that you understand how neurons creates this potential energy that we call membrane potential. If it's at rest, it's called a resting membrane potential. And that energy is expressed in volts, and it has a value of minus 70 millivolts for neurons. So this is a mini battery rated at minus 70 millivolts that can perform cellular work for neurons. Okay. Now, if you change the value of this potential, what we call resting membrane potential, right? If we change that, if we disturb that, if we perturb that, if we modify it, then it can be used as a signal. And this is exactly what we're going to talk about. The texture of the nervous system, the heart and soul of the nervous system is signaling, meaning its ability to send out signals. And there are electrical signals and there's chemical signals, right? But today the focus is on electrical signals. And when we talk about electrical signals, there are two types of electrical signals. The action potential. This is a marvel of nature. And because of this, we're gonna reserve it for the next recording, because by the time you get to the next recording, you have all the foundation under your belt to understand the concept covered by action potential. And the other type of electrical signals is the graded potential, which is the remainder of today's lecture, today's recording. So for graded potential, there are three types. There are the receptor potential, the generator potential, and the postsynaptic potential. So three types. For today, we're going to focus on the postsynaptic potential. We're going to come back to the receptor potential and generator potential when we discuss the senses. When we discuss the special senses, for example, okay? So again, graded potential, specific example of electrical signals, and the graded potential is further subdivided into three subtypes: the receptor potential, the generator potential, and the postsynaptic potentials. Graded potentials are used for short distance communication. The reason is because the graded potential lose signal intensity proportionally to the distance being covered. So the longer the distance, the graded potential can basically lose all of its signals. So that's a problem with graded potential. It cannot travel long distances because it will lose its steam. It will lose its intensity. That's why you see here, if you start out on the y-axis, that's the intensity of the signal. And as you move farther away from either side, you see that potential drops ultimately down to zero. So that's the distance, right, of travel. So when it is being generated at its location, let's say on a dendrite of a neuron or the cell body of a neuron, and it starts to spread out, as it spreads out, if it's at the dendrite, it's going to travel toward the axon hillock of a neuron. It has to go there. So that's its objective, its destination of all graded potential, especially the postsynaptic potential. Once it is generated, it has one singular objective, and that singular objective is I need to get to the axon hillock of the neuron. The axon hillock is a delta shape at the cell body of the neuron. It connects the cell body to the axon of the neuron, right? And that axon hillock is the most important location of a neuron. It is the CPU of that neuron, central processing unit of the neuron. It's like a mini brain of the neuron, because that axon hillock can decide whether or not to fire an action potential. We're going to discuss next lecture. Okay. So that postsynaptic potential, once being generated, needs to get to that axon hillock. The problem is, the farther away it is located from the axon hillock, its chance of getting to the axon hillock diminishes proportionally to the distance between the location of the dendrite and the axon hillock. So because of that, graded potential are only for short distance communication. Now, your book used the word conduction. For the entire class, change this word here, conduction, to another word. So change that conduction, replace conduction with the word propagation. That's a proper terminology. Because when we say conduction, we are talking about electrons running along a surface, running along a wire. That's conduction. In biology, if you allow that to happen, like I said earlier, those are free radicals, right? For example, high energized electrons seen during aerobic cellular respiration, that's electron conduction. It's deadly. In the case of neurons, in the case of excitable tissue, the mechanisms used to generate electrical signals, it is not relying on electrons. You've seen it so far up to this point. They rely on electrolytes, positively charged chemical, sodium and potassium. We're not talking about electrons here, so we have to change the terminology. This is short distance propagation, right? Okay. So graded potential dissipates with distance. So signal amplitudes disappears with distance. So the longer the distance for travel, the less the signal amplitude. Ultimately, it can go to zero even before it gets to its destination. Action potential, on the other hand, is for long distance, both short and long distance, because the signal amplitude does not dissipate with distance. This is phenomenal. The action potential, its ability to maintain 100% of its intensity. One, it is being born, one is being generated at the axon hillock, and when it arrives at its destination, is still 100%. It doesn't lose anything in terms of intensity. Okay, so that's the advantage of the action potential. But the action potential is hard to generate. That's why there's something called all or none principle. So either you generate an action potential or you don't. You cannot generate an action potential at 50% of its intensity, at 75% of intensity, at 25% of intensity. It's not possible. It's either 0 or 100%. So it's either no action potential at all or one full-fledged action potential. Okay, that we call it all or none principle. And that we're going to cover that on the next lecture, because the axon hillock is capable of satisfying this all or none principle. And when the action potential is, generated and propagated, and when it's propagated, means it travels, spreads out through an axon, it relies on a positive feedback mechanism. So this is self-sustaining. That action potential is amplified by positive feedback mechanism. And again, we're going to talk about it on the next one. So the advantage of an action potential is it does not lose intensity. It is perfectly designed for long distance propagation. The problem with that is you cannot add one action potential on top of another. So they work by themselves. They are mavericks. They are not a team player. You cannot add them up. So once it is being produced, the neuron needs to wait until it goes away before the neuron can generate another action potential. The axon hillock cannot generate two action potentials simultaneously. That's the downside of the action potential. Whereas the graded potential, even though it dissipates with distance, the dendrite can add different graded potential on top of one another to help each other out to get to the axon hillock, to prevent the dissipation of signal intensity due to the distance, right? So because they dissipate, they disappear, the longer the distance, they help each other out. Different graded potential, if they're being generated closely enough together in terms of time, they can add up together, help them to get to that axon hillock. So there are advantages and disadvantages to both, but so that's how, in this case, neurons use these two types of electrical signals to communicate between themselves. So let's focus on, like I said, the postsynaptic potentials. And when we talk about postsynaptic potentials, there are two flavors of postsynaptic potentials. There's the excitatory postsynaptic potential, and there's the inhibitory postsynaptic potential. So there are two flavors, right? Signals that excite and signals that inhibit. Why do we call it postsynaptic potential? Let's revisit the concept. You have a neuron here. You have another neuron here. Let's erase the color here, has nothing to do with the coloring. And you have microscopic space here. Right? And we call that microscopic space synapse. And the red neuron is the postsynaptic neuron. N, okay? Postsynaptic neuron. Postsynaptic, this word here, postsynaptic neuron. Neuron. The blue neuron is the presynaptic neuron. So I'm not going to write it. So P-R-E, synaptic neuron. Now, the reason why we call postsynaptic potential is because the presynaptic neuron releases neurotransmitter, and the neurotransmitter, we're going to learn later, will induce the formation of a potential on the postsynaptic neuron. So that's why these signals are called postsynaptic potential. So the neurotransmitter induces the perturbation of the membrane potentials on the plasma membrane of these postsynaptic neurons. Because of that, that electrical signal is called postsynaptic potential, because the potentials perturbation occurs on the postsynaptic neuron. Okay. Now, when we say excitatory, there are neurotransmitter channel on the postsynaptic neuron here that would depolarize the resting membrane potential, right? So either the dendrite or the soma Soma means the cell body, excluding the axon hillock, okay? So the cell body excluding the axon hillock. They have transmembrane protein. Okay, so neurotransmitter gated cation channel. These are transmembrane protein, TMP. This here, that's a TMP, transmembrane protein, that are called neurotransmitter gated, ligand gated, or chemical gated cation channels. And they're activated by neurotransmitters that are released by the presynaptic neuron. So the neurotransmitters are coming and they bind to these TMP. And what happened is these cation channels, when they open, they allow ions to flux through them to depolarize the resting membrane potential, to change the membrane potential. So to flip the membrane potential. So at rest, inside the inner leaflet is negative, but once this happens, the inner leaflet becomes positive and the outer leaflet becomes negative. So that's what we call depolarization. So this action flips the potential, right? We call it depolarize. So at rest, inner leaflet is negative, outer leaflet is positive. When it depolarizes, the polarity flips. The inner leaflet becomes positive, the outer leaflet becomes negative. Whereas there are neurotransmitters that will also activate transmembrane proteins. So these also transmembrane proteins. There are channels, but they flux now anions, so negatively charged ionic species like chloride. Whereas the cation are positively charged ionic species. So we have sodium, we have potassium, and we have calcium. So when you allow chloride influx, meaning chloride is coming in, it makes the inner leaflet more negative, right? So you are hyperpolarizing. You're making the inner leaflet even more negative than before. So in other words, you inhibit the postsynaptic neuron from generating the next step, which is generating action potential, right? So if you depolarize the membrane, you increase the chance for the axon hillock to generate an action potential. But if you hyperpolarize the membrane, then you will inhibit, you will prevent the axon hillock from generating an action potential, because you're making the inner leaflet even more negative than it was at rest. So the idea here is, if you want the axon hillock to fire an action potential, you make that inner leaflet less negative, not more negative, right? Less negative means you have to depolarize it. More negative means you hyperpolarize it. So if you make it more negative, forget it. The axon hillock will never find action potential. But if you want that axon hillock to find action potential, you better depolarize the membrane. You better make it less negative. Okay, so let's move on to the details on the next slide. I think I wrote down everything for you. Okay, so there are excitatory and inhibitory neurotransmitters. When we say neurotransmitters, they are released by presynaptic neuron, okay? So neurotransmitters are chemical messengers used by neurons, used by the nervous system. And in this case here, when we talk about postsynaptic potential, either EPSPs or IPSPs, EPSP stands for excitatory postsynaptic potential. IPSPs, inhibitory postsynaptic potentials. The little s here for the plural. So one EPSP, two or more EPSPs, one IPSP, two or more IPSPs. Now, if the postsynaptic neuron receives these neurotransmitters, glutamate, which is one of 20 amino acids used to build proteins. But in this case, this amino acid is being used as a chemical messenger. So it is acting a bona fide neurotransmitter. The next one, the acetylcholine, that's another neurotransmitter. Serotonin is another neurotransmitter. And ATP, the same cellular energy molecule, in this case, is used as a neurotransmitter. And actually, my PhD thesis, that was my work working on ATP-gated ion channels involved in pain perception. So I did clone a brand new ATP-gated ion channel. That was way back in the 90s when I did my PhD thesis. So that's an example. So ATP being a neurotransmitter. So when glutamate, ACH, serotonin, or ATP binds to these channels, so glutamate, ACH, serotonin, ATP are produced and released by the presynaptic neuron. They go into the synapse or synaptic cleft through simple diffusion. They migrate, they leave the presynaptic neuron, going on to the postsynaptic neuron. And on the plasma membrane, on the dendrites, thousands of dendrites, or the cell body of the postsynaptic neuron, you have these neurotransmitter-gated ion channels where these neurotransmitters can bind. And once they bind, they open the channel, and you have sodium influx, meaning sodium is moving in, calcium influx, calcium is moving in, but you have potassium efflux, potassium is moving out, right? But there's much more influx than efflux. So here we will have a net influx of positive charge. So in other words, the plasma membrane will be depolarized. You are making the inner leaflet of the membrane less negative, right? Because it accumulates a lot of positive charge. So you're flipping the polarity now. Once it happens, the inner leaflet becomes more positive and the outer leaflet becomes less positive. And this, it will lead to EPSPs on the postsynaptic neuron. So on the plasma membrane of dendrites and the cell body of the postsynaptic neuron, we have the formation, the birth of EPSPs. These are greater potential that will try to get to the axon hillock of this postsynaptic neuron. Similarly, the same concept, there are also inhibitory neurotransmitters. We have a neurotransmitter called GABA. We have another neurotransmitter called glycine. Glycine is one of 20 amino acids, and glycine is the most simple amino acids, right? Glycine is also the only one not having a chiral center. So there's no enantiomers in the case of glycine. There's no right and left hand mirror images for glycine. In this case, glycine is acting as a bona fide neurotransmitter. And not only that, GABA is released by neurons in the brain. Glycine is released by neurons in the spinal cord. So both GABA and glycine are produced and released by presynaptic neuron. They're released into the synapse or synaptic cleft, again by simple diffusion. They diffuse out, they leave the presynaptic neuron, and they bind to their receptor on the cell membrane, on the plasma membrane of the postsynaptic neuron, specifically at the dendrites, thousands of them, or the cell body of the postsynaptic neuron. When I say cell body, in this case, excluding the axon hillock. And when they bind to these anion channels, they open the pore, the tunnels, the passageway, chloride goes inside the neuron. So there's a the chloride influx. What happens here is the membrane becomes hyperpolarized. The inner leaflet now becomes even more negative than before. So you are creating IPSPs. Okay, so you are basically preventing that axon hillock from generating an action potential. So graphically, how do we express that? So here what you have is, those are the different signals. So over time, W, X, and Y. So the W here is an EPSP signal because it goes, this is minus 55, that's the threshold. That's the potential required for the axon hillock to find action potential. So this here, this would be at rest, and that would be minus 70 millivolts, right? So the W signal, this is a depolarizing signal, the EPSP, because it moves closer. It's less negative, so it's more positive, it moves closer to the threshold. This is also an EPSP. But if you look at the Y signal, it moves farther away from that negative, it's more negative, so that's an IPSP. So this is IPSP signal. And That's an EPSP. This is IPSP. So it goes farther away from the threshold potential. And this minus 55 number here threshold potential. So that's the requirement. You need to get to threshold potential before the axon hillock fires an action potential. And then the action potential, when we talk about action potential, this is generated by the axon hillock. Connecting the cell body to the axon. This is the birthplace of all action potentials. So if the axon hillock, the cell membrane, right, the plasma membrane of the axon hillock displays a value of minus 55 millivolts, minus 54 millivolts, it will fire an action potential. But if it has a minus 56 millivolts, it stays silent. It remains silent. It won't fire an action potential. And that's important. So if it's minus 55, it's okay. Let's see up here, minus 54, it's okay. It will fire an action potential. But if down here, for example, let's say this is minus 56, then nothing happens. No action potential. It has to be minus 55 or more positive. Minus 54, minus 53, minus 52, all of it is for pro-action potential generation. But minus 56, minus 57, minus 58, no, it won't happen. Okay, so now this is the dissipation distance here that we talked earlier. If you look closely on these graded potentials, right, they get generated and they die out. They get generated, they die out. They get generated, they die out. Same thing here, this gets generated and it dies out. But if these two here, they were created close enough in time, they can add up one another, they help each other, and they can get to, when they get to the axon hillock, they are above threshold, the axon hillock fires an action potential. Right, same here. This fires by itself, it dies out. It fires by itself, it dies out. But this here is multiple ones firing. So two of them firing together. This is, they add up here to get to threshold. This, they add up to get to threshold, right? So this is EPSP's graded potential ability to be added up on top of one another, help each other out to get to the axon hillock, get to the threshold potential, so the axon hillock can fire an action potential. So here's the opposite. It goes away from the threshold. So the probability of that axon hillock to fire an action potential is 0 probability. No chance. So on that note, I'm going to terminate today's recording and I will see you on the next recording, finishing off this part 2. Until then, be safe out there, be happy, study well, make me proud. Until next time, take care.